Okay, so this is the genetics and heredity review and practice video lecture, and we're also introducing a couple of concepts: codominance and incomplete dominance and sex-linked traits. So let's get started. Gregor Mendel, he's the father of genetics, just like Charles Darwin would be the father of evolution, and Watson and Crick would be the fathers of the structure of DNA, and let's not forget the mother, Rosalind Franklin. But Gregor Mendel was a priest. He lived in a monastery. He was also a scientist, and in the monastery he was responsible for the gardens, and he used pea plants to study the patterns of inheritance. Gregor Mendel laid the foundations for the study of genetics, okay? So you need to know him. Genes, nothing but genes. All of our traits are coded by genes, and genes are just little segments of DNA, right? And there can be different versions of the same genes. That was my bad attempt at a segment of DNA. Genes can have different versions, and when there are different versions of the same genes, it's called an allele, okay? And you get genes from both parents. So let's say that mom has genes for red eyes and dad has genes for blue eyes, and we get a gene from mom and a gene from dad, and they're two different alleles. This is for red color, this is for blue color, and I get a gene from mom and a gene from dad, and then I have red color. So different versions of different genes are called alleles. All genes are either dominant or recessive, according to the principle of dominance. If you have at least one dominant allele, the dominant trait will be expressed. Dominant traits are represented by capital letters, and recessive traits are represented by lowercase letters, okay? The principle of dominance says exactly that. Genes can be either dominant or recessive, and if there's at least one dominant allele, if there's at least one dominant allele, the principle of dominance states that traits can be either dominant or recessive. Dominant traits are represented by a capital letter. Recessive traits are represented by a lowercase letter. And if you have at least one dominant allele, then the dominant trait will be expressed. So say we're talking about hair color. Let's say that blonde hair is recessive, so it's going to be represented by a little b. And brown hair is dominant, so it will be represented by a big B. So dominant traits represented by capital letters, recessive traits represented by lowercase letters. Remember, we have two alleles for each gene because we get one from each parent. Let's say we have a kid that has these two alleles. Would they have dominant hair color or recessive hair color? Well, because there's not at least one dominant allele and there's two recessive, they would have recessive hair color, which would be blonde. Let's talk about the difference between genotype and phenotype. The genotype is the genetic makeup of an organism. That's like GG or AA or DD or BB. The alleles that are represented by letters, that's the genotype, okay? And the phenotype is the physical traits that are expressed. So red hair or blue eyes, or let's see, we could have long legs. I don't know if that's a genetic trait, but you get it. The physical traits that we can see are the phenotype. So somebody says, what's the genotype for that red-haired girl? It might be heterozygous, big G, little g, okay? Let's talk about how we can determine what the phenotype is based on the genotype. So here's our key here, all right? The purple flowers are dominant and the white flower color is recessive, okay? So if we have two dominant alleles, if the genotype is homozygous dominant, then we will have a purple flower, remember? Because the principle of dominance says if there's at least one dominant allele, that will be expressed, and the dominant color is purple, so this will be purple. What if our genotype was heterozygous? What would our phenotype be then? Well, there's at least one dominant allele, so the dominant trait will be expressed, which is purple. And here you can see there are no dominant alleles. It's two recessive alleles, homozygous recessive. So would the phenotype for this genotype be purple or white? It would be white. Okay. Now let's practice determining the genotype based on the phenotype. So it is dominant to have no dimples, and it's recessive to have dimples, which means you have to get two recessive alleles in order to get dimples. Well, there's two possible genotypes for a human that has no dimples, okay? Remember, principle of dominance. If there's at least one dominant allele, then that will be expressed. So let's say we have at least one dominant allele, we'll have no dimples. So our second allele could be recessive. Or we could have one dominant allele and another dominant allele, okay? The only way that we get dimples is if there are no dominant alleles, because as long as there's a dominant allele, we'll have no dimples. So our genotype for dimples has to be homozygous recessive, okay? Now, we can predict what offspring might look like based on probability using a Punnett square. So let's say someone with curly hair wants to know what their kids would look like if they had a baby with someone that had straight hair. Okay? So first let's determine what their genotypes are. Straight hair, we could have straight hair is dominant. So if someone has curly hair, they can't have any dominant alleles. They can only have recessive alleles. So this person has lowercase h, homozygous recessive as their genotype. And the person with straight hair could have one of two genotypes. It could be capital H, lowercase h, heterozygous, or it could be capital H, capital H. Okay, so let's go with capital H, lowercase h. So the way that we set this up is we take the genotypes of both parents and we put it on this Punnett square. It could be one parent on the top and one parent on the side, or vice versa. Let's do it this way. Let's do homozygous recessive, our curly-haired person on the top, and let's do heterozygous, our straight-haired person on the side. Okay, now for each box you take an allele from the top and an allele from the side. And in this box we'd take an allele from the top and an allele from the side. And in this box we'd take an allele from the top and an allele from the side. And in this box we'd take an allele from the top and an allele from the side. Okay? So we would get capital H, lowercase h, capital H, lowercase h, lowercase h, lowercase h, lowercase h, lowercase h. Okay? Let me do that again without the arrows on there, okay? So we would take this h on this side and put it in here, and this h right here and put it in here. And then we'd do the same down here. We would take this h and put it in here, and we'd take this h up here and put it down here. Same over here. One from the left, one from the top, one from the left, one from the top. Okay? Now, what were the possible genotypes in our last cross? Well, we had let's see, heterozygous, homozygous recessive. And let's actually use a different color here. So how many different possible genotypes did we have? Well, we had two, heterozygous and homozygous recessive. So we had capital H, lowercase h, and lowercase h. And we had 2 out of 4 of each of those, which 2 out of 4 is a half. 1/2 is 50%. 50%. Okay. Let me fix my 50% over here. 50%. Okay, now what were the possible phenotypes? Well, we know our possible genotypes. Our possible phenotypes were, well, this one has at least one dominant, so it would be straight hair. And this one has no dominant allele, so it would be curly hair. And the numbers are the same. There were 2 out of 4 of each of them, so the numbers would be 50% and 50%. Okay? Now, we can determine the probability of how we're going to inherit 2 different genes with something called a dihybrid cross. So, if we cross a white rat that has black eyes with a brown rat that has black eyes, what would our babies look like? Well, the first thing we have to do is determine our alleles, okay? So we take a white rat, and actually I'm going to use a different letter than F. Instead of F, let's use G. Let's use big G and little g here, okay? And I want to fix my little g just to make sure that we're all on the same page. All right, so let's cross a white rat, and remember we're using g's, so that'll be big G, little g, with black eyes, with a brown rat, no more f's, little g, okay? That has black eyes. So let's look at our first parent genotype. It would be big G, big B, little b. Okay? Now, we have to determine what the gametes are for this parent, and we do that with a method called FOIL. And that stands for first, outside, inside, last. So you have to you have to figure out how many different ways we can combine our alleles. So big G can cross with big B, that's the first alleles. We'd get big G, big B. Outside alleles would be big G, little b. Okay, the O stands for outside. The inside alleles would be little g, big B. And the last alleles would be little g, little b. Okay? Now, there are different ways that you can think about this. Let me show you a little different way with our other parent, who was little g, big B, little b. Okay? You can think about these sets of alleles independently. So the G's will represent fur color and the B's will represent eye color. And how many different ways can I pair up each allele? Well, I think about it this way. I can pair up this little g with this big B and this little b. So I would write them out. Little g with the big B and little g with the little b. And then I can use this little g with either the big B or the little b. So I would do little g, big B, little g, little b. Okay? It's a little different way of thinking about it, but you get the same thing. Okay? So once I have all of my alleles, here's 4 and here's 4, or those are gametes, all my different combinations of alleles, I can plug them into like basically a giant Punnett square. Okay, so big G, big B, big G, little b, big G, big B, big G, little b. I also had little g, big B, little g, little b, little g, big B, little g, little b. Okay, that's my first set of alleles. My second set of alleles would be little g, big B, little g, little b, little g, little b. And it's actually the same thing, little g, big B, little g, little b. If I'm correct, yes, I was correct. Now, I have my Punnett square set up, so now I just plug and chug. And remember, you take your alleles from the top and your alleles from the side. So this box would be big G from the top, big B from the top, little g from the side, big B from the side, okay? And you notice a couple of things. One, I always wrote my dominant alleles first, and I also always wrote my alleles together. I kept my G's with my G's and my B's with my B's, right? So those are two formatting rules for you. Let's go to the next box. We'll take from the top, and then we'll take from the side, remember? Okay? So big G, big B, little b, right? This one would be little g, big B, big B. This one would be little g, big B, little b. And I'm just going to keep going. Big G, big B, little b. Big G, little b, little b. Little g, big B, little b. Little g, little b, little b. Little g, big G from the top, big B, big B. Big G, big B, little b. Little g, big B, big B. And remember, I'm bringing them from the top, and I'm bringing them from the side. Okay, that's all I'm doing, top and side. Little g, big B, little b. Big G, big B, little b. Big G, little b, little b. Little g, big B, little b. And little g, little b, little b. Okay, so I filled out my whole box. Now we can go through and say, well, what percentage were this genotype and what percentage was the phenotype white fur with black eyes, etc., etc. But I am not going to do that in this particular review video, okay? So now we're into the new stuff. Incomplete dominance is when some alleles aren't completely dominant, and we see an expression of both of the alleles, a combination of both of the alleles. So in incomplete dominance, the heterozygous phenotype lies somewhere between the two homozygous phenotypes. What are the two homozygous phenotypes we're crossing here? Big R, and little r, little r. Homozygous dominant, homozygous recessive, where it looks like dominant is red and recessive is white. Well, when we get the heterozygous genotype, which we have all four are heterozygous here, we get a combination of red and white. We get a combination of red and white, which is pink. Okay? So in incomplete dominance, we get a combination of our two phenotypes, and they blend together, and we end up getting a combination of both. In this case, it was pink. Red and white made pink. That's incomplete dominance. Codominance is when both phenotypes are clearly produced. So, for example, in chickens, the alleles for black feathers is codominant with the alleles for white feathers, okay? So you see black and you see white feathers. You see them both. So heterozygous chickens express clearly black and white feathers together. If they were incomplete dominant, it would be a blend of black and white, so we'd have a gray chicken. But no, it's codominant. We see both alleles. If the flowers were codominant, then we would have a flower that would have both red petals and white petals. Okay? You see both alleles. You always see both alleles independently. Okay? So, blood type is an example of codominance. So, if I have the genotype AA, then I would have the blood type A. Okay, I could also have AO here. I would have the blood type A. If I had AB blood, my genotype would have to be A and B. I see both A and B being expressed here. For B blood, I would have to have B, B. Both would be expressed. I could also have BO here. And for O type, I have to have OO. Okay, that is expressed. Okay, and that is all for this presentation.